also in the International Conference on Mathematical Neuroscience in 2022. In my talk, I will introduce some of the ideas that we have been developing within Neuromat, and I take the opportunity to acknowledge my funding agencies and the support of FAPESP to our project Neuromat. And the title of my talk is Learning and Compressing Stochastic Sequences of Events by the Brain. So this project is enrolled in the research line called the statistician brain. It is a Neuromat research project. The idea that the brain makes statistics so as to interact in the world. This idea is by no means new. Predicting means anticipating outcomes. This proposition was done by Hermann von Helmholtz, a multi-scientist from the 19th century who proposed that the brain would predict, and by predicting would learn regularities from the world. This model has been adopted by many scientists in the 20th century as the Helmholtz heritage. In the 20th century, some other very eminent neuroscientists, such as Marc Janot, took over this idea of prediction to reframe it within the idea of the mental simulation states within the model of motor cognition. So mental simulation states relates to pretended actions, imagined actions, prospective action judgment, action prediction, and action observation, as well as action in dreams. In all these states, we expect, Janot proposed that there will be an activity in the brain that associates to simulating the execution of these actions, and thereby creating estimates of outcomes. Today, our proposal is to discuss with you the more in depth this concept. So how does the brain assign probabilistic models to sequence of stimuli so as it learns to act in the world? Take, for instance, our world champion in pole vault, Fabiana Müller. She has developed through her many years of training models of how to behave, how to interact, how to produce this very skilled movement so as to execute these actions in an exquisite fashion. In the lab, to investigate this action production, we have to work with reduced information, so that, for instance, when you put markers in each joint and retrieve the information that associates to a person walking, such as the one that I'm showing here for you today. This has been done in several labs, and we took this type of approach to investigate the brain activity that associates to the visualization of these types of stimuli. So here is the type of experimental setup that we employ. A person is sitting and watching these videos depicting these movements I just showed you, while we record the brain activity in specific points of the skull. And this activity can then be analyzed, for instance, by extracting graphs of interactions among these electrodes in the skull. So this was done. It's a work published in 2014 by Freiman and collaborators. And this is the type of information that we can retrieve from this type of experiment. You can see here that while the participant watches the videos and we reconstruct this brain activity, there is a ever-changing activity across time. So this is repetition 1, repetition 2, and so on and so forth. And a trick in this type of experiment is to compare the biological movement that you just saw that evokes the sensation of visualization of a walking person, and to compare this type of graphs generated by the visualization of this person with the scrambled condition, where we take each of these markers and we put in a different position than from the original. So we call it scrambled motion. When we compare these two conditions, we get very interesting results on how the brain extracts information from this typical stimuli, and we can compare in a very detailed way which would be the brain regions that are more related to the identification of the human body in motion. However, very little can be extracted from the signatures that associate to each of these stimuli in the brain. So it's a broad distributed information on the correlation between electrodes, and some information on which will be the regions best suited to distinguish between the two conditions. So let's move to another paradigm, also associated to movement observation. This is a work which was done in collaboration with Angela Sirigu team in 2004. Here again, we ask the participants to watch videos of movements and no movements in the screen. So in the right, you can see the type of videos that have been shown to participants. One condition was called the movement observation condition, where a person directed the movement towards a green object. So the person knew beforehand that each time the green object was appearing in the screen, there will be a movement directed toward this object. And we compared this condition with the second condition that was called no movement observation, where the person watched the video with a still hand and a red object just in front of him. So the person knew when he watched the red object that there will be no movement in the screen. And there was a third condition called the execution condition, where the person had to press a button upon a signal or an indication. In fact, a red object would appear in the screen and the person would have to press a button. We were searching for the redness potential, a marker of motor preparation. So this is known since the 60s, that each time a person performs a given movement, there is a negativity, which is called the redness potential, that builds up just before the beginning of the movement. It's a very well-known marker of motor preparation, and it's known to recruit a large network of brain regions that associate then to the motor execution. And our question was, is it possible to elicit an equivalent of the motor preparation activity whenever the person watches a movement or prepares to watch a movement that is coming to the screen? So these are the results from this very first study. As you can see, there is a clear negativity or clear redness potential occurring before the beginning of the action, and also a negativity that associates to the green condition, meaning that each time the person is faced with the possibility of an action occurring in the screen, the person prepares by observation. It predicts the occurrence of the movement to be performed by in this case, in the video screen. And here in red, we see the brain activity that associates with the condition where there will be no movement in the screen. So these were very interesting results. And we then more recently reproduced this experiment by adding a new condition. So here in this new experiment, we had the no movement condition that you already know, and we're looking again at this motor preparation activity before the beginning of the action. The ball movement condition, the ball will go towards the hand, and the hand movement condition where the hand would go towards the ball. So we introduced this ball movement condition where there was an object coming towards the space around the hand. We recorded the electroencephalographic activity from selected electrodes in the scalp. So this is a top view showing the distribution of the electrodes of interest. In red, the electrodes above the sensory motor cortex, and in blue, the control electrodes that were disposed over the temporal, the temporal auditory cortex that would not be related to this specific task, so as a control condition. And this time we employed a different statistical approach. We used a hierarchical clustering analysis, and we compared the three conditions: ball movement, hand movement, and no movement in pairs. So this is represented here in this table, where you can see, for instance, for a control group with 9 participants, we could verify that when, for instance, we compare ball movement versus no movement condition, when the person was watching a video depicting the dominant hand view, it was possible to separate, to distinguish between these two conditions in 7 out of 9 participants for the contralateral hemisphere, and in 9 out of 9 participants when we were looking at the electrodes in the ipsilateral hemisphere, in the hemisphere ipsilateral to the viewing hand. So here again, we, to wrap up this first part of the talk, it's possible to extract from the EEG signal signatures, specific signatures that associate to S states. So we did it by building graphs of interactions from EEG signals, and we have been able to show that those that were related to human movement were different from those that were extracted from the viewing of scrambled movement. We also showed that there were specific EEG signatures associated to upcoming hand movements performed by others. And more recently, these upcoming hand movements have been shown to depart from those evoked by upcoming tactile movements, the condition where the ball goes directly to the hand. So this means that it's possible to retrieve specific information associated to the EEG signals. But at this point within Neuromath, we had some open questions. We wanted to go a step forward. How to extract from brain signals the very structure of a sequence of events. In the previous experimental protocols I just showed you, the events were scrambled. So there was no way of extracting information of the very structure of this sequence of events. And how to establish a formal relationship between the sequence of events and the recorded signal. So the Neuromath context, the team was the perfect context for us to explore these new venues in this domain. So our idea was to use a sequence as a formal model to and construct a sequence of stimuli, a structured sequence of stimuli that could be retrieved from the EEG signal. And the chosen model to produce this sequence of stimuli was the one presented in 1983 by Jean-Marie Hissening. He published this famous paper called the Universal Data Compression System, where he formalizes a system that allows compressing sequences of symbols. And this will be the detailed, this detailed approach will be explored by Aline Duarte in her talk. We decided to draw a new experimental protocol, which was published in this paper. And the idea was to employ, again, data recorded from EEG signals, but this time employing sequences of hand claps. We had two auditory hand claps, two hand claps that were employed, a strong beat, a weak beat, and the third auditory unit, which was the silent unit. And we employed two different trees, the context trees proposed by Hissening, as a way to compress this data. So we created the ternary tree and the quaternary tree, and the sequence of events are represented here. For the ternary tree, for instance, 211-201 means 211-2-1. So that the weak beat could be replaced with a certain regularity by a silent unit. And for the quaternary tree, we had a different structure where there were 2-1-0-1. So it was also called the valse, 2-1-0-1, 2-1. And here again, the one could be replaced by a silent unit. These trees are represented, can be represented below as structured as a tree where the past events are represented by the leaves. And for each of these contexts, each of the leaves, which are called contexts, we have a probability associated of occurrence of each of these contexts. This will be detailed by Aline, as I said. And what we decided to do was to associate segments of EEG to each of these contexts. So for instance, for a given past event, a sequence of past events, we would segment the EEG here, for instance, depicted in pink. If we have a sequence of 1-1-2, the segment of EEG associated with this sequence will be identified along the signal and will be analyzed, will be compared with a situation where I have a different past, a different sequence, a different context, in this case in green. So this is the approach that was employed to extract from the EEG signal, to reconstruct, to retrieve the structure of the probabilistic sequences of auditory stimuli from the EEG data, which will be presented today by Nosley Hernandez. So a second wrap up. An important step forward in the statistician brain conjecture is to establish a formal relationship between the structured sequence of random stimuli and the stochastic process associated to that brain processing of those stimuli. This was done by employing the functional data selection procedure that will be described by Aline Duarte in the next talk of this mini symposium. Nosley and Hernandez will then show results supporting the conjecture that the brain effectively identifies the context-free characterizing the source, and Marcela Svark will then propose a new procedure of clustering this EEG dataset by law. So thank you very much for your attention.
Thank you, Claudia. Thank you. Okay, we have time for questions. I don't chat. So there are 4. So in the chat, so Mehran Hossein is no, no. Okay, no, I'm sorry. No, that's not something else. So I don't know how to act to receive questions. People from YouTube cannot address questions. So I don't know exactly. Okay, so let me ask. So maybe Massi has a question, or Etienne, or Mehran.
Yes, sure. Can you hear me?
Yes, perfectly well.
Thank you, Claudia, for this talk. I'm not, maybe I'm totally wrong, but I understand that your model is static from the point of view of the connection. Am I right, Claudia?
From the point of view of the connection,
the graph is fixed at the beginning of your signal and so. And do you think it's possible to include some learning or plasticity and so in your study?
yes, thank you very much, Etienne, for your question. It is an excellent question. I think that indeed our problem in this type of experiment is that we have to reproduce, to replay these events many times, and very often you have to average these events so that you can have an information that you can, associate this specific marker of activity to that event. And so this is a limitation indeed when a person wants to investigate the learning process, right? Because we have to collapse time, so to say.
Sure.
And it's always challenging to, so one alternative is to build these windows of analysis that you can move through your data so that you can extract for each window of time, you could extract, so to say, a given pattern, and then you could compare across these windows to see if you retrieve some information that relates to learning. This is the type of approach that we've been trying to employ within neuromath. When we did the graph study, we tried to extract the modifications through time by comparing sets of graphs. And in fact, what I showed you there is actually a window. It's a graph built from a window of time. And if for each, 120 seconds, you can build a new one, and then you will try to infer if there is a signature that associates to the learning of this process. But we are still working on it, and I think it's a challenge for us to develop these tools that will allow us, because there is a limitation, an obvious limitation, which is that you cannot keep your participants forever in this doing the experiment. We have to be.
Well, I think as a chairman, I'm obliged to stop there because it's well exactly in time. but I could say that it's a very good question. We're working on this now, in particular using behavioral data in the goalkeeper game, in which you don't have the limitation you have with EEG. And I expect in the next ICMNS there will be a mini symposium to present the learning models we are just introducing now. Thank you very much, Claudia. Okay.